Bismillahirrahmanirrahim. Let's talk more about the environmental biodegradation of dioxins. Now, there are many different methods that can be employed. Those methods can also be employed in other cases of different toxicants and contaminants. For example, excavation and incineration of soils can be done. However, as we know, these are very expensive processes. Moreover, if dioxins are present in some water body, we cannot use these techniques. So obviously biodegradation is the most desirable choice in this case. Now, as we said before, dioxins are one of those compounds which are hard to degrade. उनकी degradation बहुत slow है और that takes time, and the microorganisms and the related microbial enzymatic reactions वो भी इतने खास ubiquitous नहीं हैं. However, still many microorganisms are known to bring about the conversion of dioxins under both aerobic and anaerobic conditions, depending on the condition and depending on the parameters that prevail in the site, the transformation will vary. Now, in many studies of aerobic biodegradation, only limited biotransformation has been observed in case of dioxins. Basically, again, the process is almost the same as with any other chlorinated toxic. That it involves the addition of oxygen with oxygenase enzymes. That results, in this case, it results in the unproductive formation of mono and di hydroxylated analogues. Why unproductive? Because these analogues which are produced due to this oxygenation, these accumulate with no further degradation. So हमने पहले भी बात की थी कि जो biotransformation होती है soil में या water में due to microbial processes, that biotransformation can either completely mineralize the compound, or it can convert a compound from high toxic form to low toxic form. However, it can also work oppositely. It can convert an organic compound, a toxic compound, to more toxic compound. Depends on the type of biotransformation that is happening. In some other cases, when this biotransformation happens, it doesn't always lead to complete mineralization. It can make such products which are actually more hard to be degraded biologically. Same is in this case, that the oxygenase, when it acts on dioxins, it results in the formation of mono and di hydroxylated analogues, which are actually more resistant to degradation. So they increase in concentration and they keep on accumulating. Now, many bacteria are also known to contain selective dioxygenases which can reduce to oxygen that simultaneously substitute an hydroxyl group at an ether bond carrying carbon and an adjacent unsubstituted carbon in these dioxin molecules. So if that thing happens, this can lead to cleavage of ether bonds and compound mineralization. So if dioxygenases are there, they can reduce they can do the process twofold, twice at certain adjacent points. In this case, if that happens, only then the dioxins can be almost completely degraded and complete mineralization can be observed. Now, if we talk about anaerobic conditions, then reactive dechlorination of dioxins has also been observed. इसको हम पहले भी डिस्कस कर चुके हैं in case of other chlorinated compounds. TCDD, जिसको हमने अभी कहा कि is perhaps the most toxic synthetic compound known to human beings. TCDD congener of dioxin can also be formed through reactive dehalogenation of other congeners. यही हमने पहले भी बात की कि different congeners हैं, उनमें different किस्म की biotransformation अगर हो, those can lead to the formation of even more toxic compounds. So we can see here TCDD can also be generated. Now, dehalogenation obviously is a slow process, and when it happens anaerobically, it is slower obviously. However, the potential is there because the chemical structures and the chemical compounds which are hard to degrade in aerobic conditions, those can be degraded anaerobically because microbial processes and microbial enzymatic processes are known to operate under anaerobic conditions which can degrade such compounds. Why the process is slow and why it is hard to degrade? There are so many reasons. One could be the low solution concentration of dioxins in the solution form, and their tendency to partition to sediments of soil and organic matter Therefore, it leads to reduced bioavailability of these compounds. चूँकि ये पार्टीशन कर जाते हैं सॉइल में और ऑर्गेनिक मैटर में, सो ये अवेलेबल नहीं है माइक्रो ऑर्गेनिम्स को उन पे अटैक कर सकें और उनको डिग्रेड कर सकें. These are perhaps almost the same reasons which also make the other compounds recalcitrant as well. This was about dioxins. I'll see you in next lessons.